A-Level Biology 9700, Paper 5, Variant 2, October, November 2025. Question number 1. So, question number 1 is the plantain lily, Hosta planta guinea, as shown in figure 1.1, is a flowering plant found in Asia. Leaf with stomata on the lower epidermis. The hydrostatic pressure increases inside guard cells when water enters the guard cells down a water potential gradient. So this affects the width of the stomata. Figure 1.2 shows one open stomata from the plantain lily surrounded by two guard cells. You can see in figure 1.2. Now a student investigated the effect of different sucrose solutions on the width of stomata in the leaves of the plantain lily. The student prepared 5 microscopic slides as shown in the table 1.1. So you can see the table 1.1 has been given over here on different concentration of the solutions. These are the 5. Now removed the lower epidermis from a leaf of plantain lily. Immersed one small piece of lower epidermis in the drop on microscopic slide 1. Placed a cover slip over the piece of lower epidermis. Observed the stomata using a light microscope fitted with an eyepiece reticule. So repeated these steps for microscopic slides 2, 3, 4, and 5. Now state the independent and dependent variables. So we have to write the independent and dependent variables in this slide. So you can see here in this investigation, we are going to observe the different concentrations of with different concentration of sucrose solutions, the stomatal aperture, the opening of this stomatal aperture. So the independent variable in this is the concentration of sucrose solution. And the dependent variable, which is changed after the different using the different concentrations of sucrose solution, that is the stomatal aperture, or we will write as width of stomatal aperture. Now the student prepared 20 cubic centimeter of 2.5%, 5%, and 10.0% sucrose solutions from a 20% weight by volume stock solution. Complete the description of the student's dilution method for the 2.5% and 5% sucrose solutions by writing the correct volumes in the following statements. Students, to prepare 2.5% sucrose solution, the student has to mix the 2.5 of 20% sucrose solution with 17.5 of distilled water, milliliter of distilled water, because we have to use the formula C1V1 C2V2 for this purpose. C1V1 C2V2. For this purpose, we are going to find out the concentrations. You can use this formula, and after this, by using this formula, we have come to know that we are going to 2.5 cubic centimeter of sucrose solution with 17.5, because we are using the 20% of sucrose solution for this. As you can calculate it with the help of calculator, that we are how it can be calculated. That is So it is equal to 20 because we have to volume V1 and V2, it is 20. So we have to find out the concentration of which we have to take. So according to this, the volume will be 2.5 and 17.5. And the other, it will be same as 5 cubic centimeter stock solution with 15 cubic centimeter of the distilled water. So with the help of this formula, you can find out this answer which I have calculated. Now the next part of this question is, the student observed stomata on the microscopic slides using the high power objective lens. So outline a method the student would use to investigate the effect of the sucrose solutions in table 1.1 on the width of the stomata in the leaves of plantain lily. Your method should be set out in a logical order and be detailed enough to allow another person to follow it. Details of how to prepare the microscopic slides not be included. So we have to find out the method. We have to write down the method, uh, which will completely describe the. So for this purpose, we will start as first of all, we will write down the this method as first of all, we will calibrate the eyepiece reticule using a stage micrometer under the high power objective lens to ensure measurements can be converted to micrometers. You know that in A levels, we use the unit micrometers for our calculations. Then we will equilibrate, we will allow the epidermal peels to remain in the sucrose solution for a set period uh, that can be 10 to 15 minutes to reach osmotic equilibrium before observation. Then we will select a predetermined number of stomata from each slide, use a random or systematic sampling method to avoid bias. So we will use the eyepiece reticule to measure the width of the aperture of each selected stomata. Then we will ensure the measurement at the width point. Then we can repeat the entire process with multiple epidermal peels for each concentration to identify anomalies and calculate a mean. Then the control variables will be the temperature and with a stable ambient temperature. Then we will use the peels from the same plant and the same leaf positions to ensure genetic and physiological consistency. Ensure the light intensity from the microscope lamp remains constant at light effects to metal aperture. So we can use the eye for our eye protection when we can use the goggles when handling sucrose solutions, and we can wear the gloves, or we can make use of forceps or scalpels to obtain the peels. So these are the different which we can make use over there. So we have to write all these points over here as I am going to style. This is of 5 mark. So we have to at least we can complete it within 5 points. So first of all, the first point in this regard is the calibration. So using a stage micrometer, you know how to calibrate this. under the high power objective lens. Then allow the epidermal peels to remain in sucrose solution for a set period. Select a predetermined or pre-decided number of stomatas from each slide. Then use the eyepiece reticule to measure the width of the pressure. Of each selected stomata. Repeat the entire process with multiple epidermal peels for each concentration to identify anomalies. And calculate a mean. Temperature, light intensity, and same plant peels from same plants will be the control variables. Wear goggles for eye protection and use forceps to obtain the pH. So it was the answer for this part of the question. Now moving towards the next page. Predict the effect of increase the sucrose concentration from 0 to 20% on the width of the stomata in the leaves of plantain lily. Explain your prediction. So here, first of all, we have to predict, and then we have to give the explanation for our prediction. So first of all, we can predict that is the width of the stomatal aperture will decrease. So this is the fact, because as the percentage, as the percentage will increase, sucrose concentration will increase, so the aperture will also become wide. So the width of the stomatal aperture will decrease as the from 0 to 20% concentration will increase. So in this way, uh, they we will predict that is the width of the aperture will decrease as the sucrose concentration will increase. Because as the concentration is increasing, the water pressure is lowering, so it will affect the width of the aperture. Now, water potential gradient, it will depend. For its explanation, we will say that the water potential, osmosis, and turgidity. So these are the factors which will affect the opening of these stomatas or closing of these stomatas. So the water potential of the solution becomes more negative than the water potential inside the guard cells. So water leaves the guard cells by osmosis, moving down the water potential gradient through the partially permeable cell surface membrane. So the loss of water causes the guard cells to lose turgor pressure and become flaccid. So as the pressure drops, the elastic cell walls change shape, causing the stomatal pores to narrow or close completely. So how it happens, I'm going to write the explanation. Water potential becomes more negative, or lower, you can say, than the water potential inside the guard cells. And in osmosis, water always move from higher water potential towards the lower water potential. So water will leave the guard cells. Water will leaves the guard cells by osmosis. The loss of water causes the guard cells God cells to lose further pressure and become flaccid. Next question, part B. Another piece of lower epidermis from the leaf of plantain. Lily was immersed in a large drop of distilled water on a microscope slide. Figure 1.3 shows the lower epidermis viewed using the low power objective lens. The student determined the stomatal density on the lower epidermis using figure 1.3 only. So you can see in this figure, use figure 1.3 to calculate the stomatal density on the lower epidermis. Use the equation A for area, pi r square, or pi is equal to 3.14 or 22 by 7. So here we have to, first of all, we have to find out the number of stomatas in this picture. So they are 1, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So the number of stomatas which we can see are, they are number of stomatas, that is 13. And then they have given us the diameter of this picture. We have to find out the diameter of picture, which is, you can calculate it by yourself, because it would be different on your question paper. So the calculation will be different. So it is about 85 millimeter, because according to the size of the page, it is changed. So 85 millimeter is, could be 85 millimeter. So the radius is equal to 85 divided by 2 is equal to 42.5. And you can see the length of the scale bar. They have given us this scale bar. So it is 16 millimeter. And they have given 40 millimeter. So first we have to find out the actual radius first in millimeter. So the actual radius, we have to convert this 40 micrometer into millimeter, or we can convert it afterwards. So actual radius is equal to 42.5 divided by 16 multiplied by 40, and we can convert this 40 into millimeter. So it will become equal to, radius is equal to 42.5 into 0. We will divide after calculating this. It is equal to 106.25. Now we will convert this into millimeter. So after, it is 0.10625 millimeter. So this is the calculation. You can calculate it by yourself. Now the area is equal to pi r square. So 3.14 into 0.10625, 0.1625. So it will be equal to, let me calculate, but you can calculate it by yourself. It will be better for you. 0.10625, 0.10625. So it is nearly equal to 0.03544. And then density, the number of stomatas are, so it is number of stomatal density, number of stomatas divided by area. So it is 13 and divided by, so it is equal to 0.366.8, or we can write it 0.367 millimeter minus 2. So you can calculate it by yourself as well. You will find the same calculation, or there will be little bit small difference over there. Now the next question is, next part of this question is, suggest how this investigation could be improved to increase confidence in your answer B1. So we have to for this purpose, we are going to write the answer. To increase confidence in the result, we can calculate a mean, we can check for outliers, or we can increase sample size. So any one of these you can write. Stomatal counts for multiple different fields. From multiple different fields, and take a mean. Use or increase sample size. Now, the last part of this question is, a different plant species had a lower stomatal density than the value calculated for plantain lily in B1. Suggest how the environmental conditions of the different plant species may vary from those of the plantain lily. So you have to explain your answer. So here we have to discuss this condition, that is the different plant species likely lives in hotter, drier, or windier environment. That is, they live in different environmental conditions. So having fewer stomata reduces the total surface area available for water vapors to diffuse out of the leaf. So this is our survival strategy to conserve water in habitats where water is scarce or where the rate of evaporation is very high due to high light intensity or low humidity. So the different, you can say, they can adapt themselves due to high light and density. Often, they increase energy for gas exchange or low humidity to prevent excessive dehydration or high carbon dioxide. Lower density, the plant can get enough carbon dioxide with fewer openings. So we can write these 3 points which I have discussed. Having few stomata reduces the total surface area for water vapors to diffuse out of the leaf. This is the survival strategy to conserve water in habitats where water is scarce or where the rate of evaporation is very high due to high light intensity or low humidity. So it was all about question number 1. Please like, share, and subscribe my channel. Thanks for watching and wait for the second question.